Whether you are a biohacking enthusiast, a plant designer, or a lab rat in biotech field, there is no doubt that you've seen this device many times. It's very powerful yet very simple device, a gel electrophoresis machine. It's a straightforward and quite simple device which has done science field a huge favor in terms of simplicity of many protocols. If you're just about to start your journey in synthetic biology and gene editing, you better learn how this device works from head to toe because it's gonna become your best friend in any required gene analysis and digestion. So let's go through how it actually works and how you can utilize it on daily basis. Let's say you cut up your piece of plasmid which contains a GFP gene and you want to isolate only the region which codes for GFP and discard the rest. You load your DNA into small pores made by a comb, run the gel, and after a few hours you get this separation on your gel. Since we know that GFP coding sequence is smaller compared to the whole remaining plasmid, we can accurately say that the DNA that has gone farthest is our GFP sequence. The gel itself is made out of a low melting point polysaccharide called agarose. After it sets in cat, solidified agarose forms many pores, which enable DNA to move towards the positive charge, and that makes agarose so attractive for DNA screening and isolation. For the run itself, you need a high voltage DC power supply, which will consistently power the device for hours, and a buffer in which all this takes place is TAE buffer. Well, Tris acetate EDTA buffer is very commonly used in electrophoresis because it maintains the pH level and is also a good electroconductor. pH level maintenance is important because it protects the DNA from degradation. A few more details that we're missing here is the fluorescent marker, DNA dye, and DNA ladder for control. Well, you can't really see the DNA, can you? Let's say you load the naked DNA in your device and run it. Even if it does go through the agarose mesh, you won't really be able to see it, will you? So for that reason, we use the fluorescent marker called ethidium bromide. We gotta mention though that it's very toxic and quite carcinogenic, so no physical contact, and basically the well ventilated area is a key for safety and for basically staying alive. Duh. On the other hand, the DNA dye is just a dye, it doesn't emit fluorescence. The dye only gives the DNA some color so we can better visualize the pipetting process. And finally, DNA ladder is just a reference. You order it from the biotech company and you know beforehand that the ladder's length is certain base pairs. So by the comparison, you can tell how long your construct can be. But the last but not least, the industrial brand new electrophoresis devices are quite costly, but the reality is that you can very easily DIY it. The coolest thing in biology is that the parts of its devices are easily DIYable, including the thermocycles for PCR, electrophoresis machine, a cell incubator, a centrifuge to some extent, and many more, so you can always find the alternative. Finally, hope you find this video helpful and see you next time.